It's a great pleasure to be here today, and what an incredible talk to follow. Thank you, Liz, for the amazing insights into what's happening at the synchrotron. Really fantastic work. What I'm hoping to do today is to talk more about what you can do with the lab-based tools. So not everybody has access to a synchrotron, and actually the complementary nature of what happens in the lab is often very useful to understand. So I'm going to be talking about some of the work that people are doing with the X-ray tools that you can have sited in your own lab. But before starting with that, let's go back to the really beginning. So why would you want to use X-rays at all? And it's this access to internal structure without physically having to section that makes this technology space so exciting. This mouse kidney, and Liz showed wonderful examples in the previous talk of being able to reach this kind of information without physically having to section the specimen. And that's really what's so tempting, and how this technology complements other imaging technologies that you might have available at your fingertips already. And from the technology perspective, X-ray imaging is extremely varied. Everything from clinical scanning over on the left-hand side of this graphic, where you're imaging whole people, to what you can do imaging rodents, perhaps in the lab, looking in vivo at how rodents are growing and internal structures are changing. More to the lab-based benchtop systems, so micro-CT and X-ray microscopy, which is where the bulk of what I'm going to talk about is going to focus today, right the way through to the nanotomography resolutions that you can achieve at the synchrotron, or indeed on lab-based tools as well. So it's an incredibly varied technology space. And it also, as I said, complements many other imaging technologies that are available as well. And to kind of illustrate that point, I thought I would take this example, which is actually from Todd Schoberg, who is at the University of Wyoming. He works on the Drosophila fruit fly, and he has many different technologies in terms of imaging at his fingertips, which are used for different things. And if we think about the imaging space generally, whether you're wanting to look at atoms or whether you're wanting to look at whole organisms, there are so many different technologies that you have available. So electron microscopy for really high resolution insights. You've got medical CT and MRI also, when you're looking at millimeters to meters, and light microscopy kind of sits in the middle. And it's the complementary nature and the complexity of the tissue and the specimen that you want to explore, which really define which of these technology types you would choose. If we bring that into the length scale of the Drosophila fruit fly here, for example, if we look at what you can do with light microscopy, incredible live cell imaging, looking at neural stem cells in this case, dividing to make the brain in that Drosophila fruit fly live. Anybody who's used a light sheet system or seen a light sheet system may also have come across images like this, where you're looking at the development and the lineage of the cells in the developing embryo of the Drosophila fruit fly. But at some point, light microscopy just becomes a bit limiting. And that is particularly true if you think about the idea of actually imaging the whole fruit fly. So rather than parts of the fruit fly or the fruit fly embryo, if we're looking at the whole embryo at the macro scale, sometimes techniques like light microscopy can become more difficult to optimize. So for example, this is a really carefully dissected abdomen taken from the Drosophila fruit fly here. And using a stereo microscope, you can kind of pick out some of the parts of it, the hindgut, the sperm pumps available, you can see the accessory gland, but there's not a huge amount of information that you can see. And that's when Todd uses the power of X-ray technology to be able to look at exactly the same specimens. This is another intact abdomen that's been isolated from the fruit fly. And using X-ray technology, you can get to access to all of these structures inside the abdomen without physically having to section at all. And for his work, it's the complementary nature of all of these different technologies, so-called light sheet, electron microscopy, and X-ray technology, that really gives him access to the information that he needs to address his research questions. So if we think about how you actually capture an X-ray tomography image, whether you're at the synchrotron or whether you're in a lab-based system, the basic technology is the same, and Liz already did a great job of introducing how that works. Essentially, you have a source of X-rays, you have some kind of detector, and your specimen sits in the middle. And in lab-based systems, your specimen turns, and you capture a series of these two-dimensional projection images. And you then use those two-dimensional projection images, hundreds or even thousands of them, to be able to generate or calculate your three-dimensional internal structure. So that's the basics of how you would actually do the capturing itself. But there are different ways of being able to generate magnification in your specimen. So in a micro CT system, which is in a lab-based micro CT system, instead of having the synchrotron as your source, you have a point source, and that produces a cone of X-rays. and your specimen sits in that cone of X-rays. And if you move your specimen closer to the source of X-rays, that's what can generate an increase in resolution. So in this case, if we move the apple closer to the X-ray source, we see an increase in resolution. We can start to look at the seeds inside that apple. And that is called geometric magnification. And it works beautifully well. It's very dependable and gives great results. But at some point, the physical size of the apple or whatever sample it is you're looking at means you physically can't get that sample closer to the source. And in those cases, you need to invoke the help of something else. And one way of doing that is by using objective lenses, like in a light microscope, scintillator coupled objective lenses, which enable you to be able to increase magnification by changing the objective lens. So in the X-ray microscope, as opposed to the micro CT system, you have your geometric magnification, but you combine that with this optical magnification. And it's this two-stage magnification that means that you can get to higher resolutions in your specimen, which, if that's what you need, is the only way of being able to do that. So this is the kind of the guts inside the system. Here are the objective lenses. Anyone who's used a light microscope before, this would look very familiar. Each of these objective lenses has a different magnification and a different scintillator coating. And essentially, you can start with one magnification and move to the next one, depending on how much resolution you actually need. And then when you look at the sort of resolutions that you can achieve, this is a bone sample here. The X-ray microscope image is shown here, and you can make out the osteocyte lacunae. You can start to quantify where they are. Whereas with the micro CT system, with just the geometric magnification, it's more challenging to be able to do that. You don't get to those kind of higher resolutions. And these multi-scale workflows really are the reason why the X-ray microscope comes into its own. This is a zebrafish specimen here. And again, if you've used the light microscope, it's quite a familiar workflow. You start by imaging a large field of view, you image the whole fish at lower resolution. You then identify the regions that you want to capture at higher resolution, change objective lens and zoom in. So in this case, it was the ear of the zebrafish and the heart of the zebrafish that were particularly interesting for this researcher. And by using this multi-scale workflow, you can get to cellular resolution, but within the context of the whole fish. And so much of what we talk about with X-ray microscopy is that it's really high resolution, but in the context of the larger specimen, which is what brings so much power to some of these workflows. So from the technology perspective, from the ZEISS perspective, we have 3 different X-ray tools available. We have our micro CT system, which is our X-radia context system, which will get you to below a micron in terms of resolution. The X-radia Versa is our X-ray microscope, and much of what I'm going to focus on application-wise is based around that tool because it's just incredible lab-based technology. That will get you down to 450 nanometer resolution. And then we also have a nanotomography system, which is the X-radia Ultra, which will stretch down to 50 nanometers. Much smaller specimens are required for that, but very cool technology nonetheless. As I said, most of what I'm going to focus on will be around the X-ray microscope tool. So for life sciences, X-ray imaging is not something that's been fundamental for many years. It's definitely an area which is growing, and what we're seeing is that there are more and more micro CT and X-ray microscope systems in facilities, which, because of the complementary nature of this technology relative to other microscopy types. I guess the bone and mineralized tissue world realized the power of what you could do with X-ray imaging many years ago, because bones are quite challenging to image with other microscopy techniques. They're hard to section, they're relatively quite dense, and actually using CT, you could just image straight through without having to physically section. So there's been a lot of work done within the bone and mineralized tissue world around X-ray imaging. But read areas like developmental biology, soft tissue and organs, so organoids, looking at whole organs, looking at cancer research, for example. Those areas are increasingly using this technology to be able to address questions. There's a whole body of exciting stuff happening within the plant sciences world, and I will show you a bit of that as part of this presentation. The world of natural history, obviously specimens that are millions of years old, completely irreplaceable. You can't be chopping those into bits, so you need to be able to image those non-destructively. Implants and tissue engineering. How well does your implant actually integrate with the new bone when you start to implant it inside the specimen? You need to be able to look at those interfaces non-destructively, and X-ray technology offers advantages for that. And also, again, as Liz alluded to, the idea of using an X-ray tool as part of a multimodal imaging workflow. So as a step in a multimodal imaging workflow, we have an increasing number of people who are using X-ray technology for that purpose. And when we talk about cellular resolution, this is a really lovely example of putting something in and seeing what you could get. This sample was prepared for volume EM. It's HeLa cells, which have been mounted in resin and they've been osmium stained, ready for volume EM. And the idea here was just to put the sample into the Versa system and see what kind of resolution is achievable. And as you can see here, you can even get to subcellular resolution using the X-ray tool. The sample is small, so you have opportunities to be able to do that. But just to give you an idea of scale, you can look at single cells or subcellular, and you can look at things as big as footballs. So this is a technology which is extremely varied in terms of the sample type that you can place inside it. But it's not just about resolution, and this is a key thing, again, highlighted in Liz's talk really beautifully. In life science samples, they're really low absorption often. There's not much there to be able to image, so you need to have good contrast. And the X-ray microscope is really optimized to be able to give you the best possible absorption contrast. Each of those scintillator coatings on the objective lens is optimized for a particular energy range, which gets you the best contrast possible for each of those objectives. So this is a mouse embryo here that's paraffin embedded. It's completely unstained, and it's been imaged using the flat panel detector here in the first instance, so we could image the whole embryo. And then 2 different regions of the specimen were imaged with the 4X objective. So we changed to a different detector, a different objective lens to get to those high resolution insights. And for this specimen, it wasn't so much the resolution that was the key thing, but it was the contrast. And for life sciences, so often that's the case. You need offered access to both of those parameters to get the most out of your specimens. And propagation phase contrast is another way of being able to do that. If there really isn't sufficient difference in absorption contrast in your specimen, you have to try and find other ways of being able to generate contrast. Propagation phase contrast relies on a difference in X-ray refractive index between different components of the specimen. So in the top left-hand image here, this is a pear sample, so a fruit sample that's been imaged using propagation phase contrast. There's a difference in refractive index between the walls and the cells, and that allows you to be able to pick out this network without physically having to put any staining or anything into that specimen to create more absorption contrast. Next, on the right-hand side, this is a fly in amber, so an example of a very old specimen. Without using propagation phase contrast, you really don't see much at all. This is just a very, very old fossil and you can't see the fly. But using propagation phase contrast, you're able to pick out the membranes of that fly and you can actually see that structure. So it was kind of essential for this study as well. Another example at the bottom, this time the challenge was that the goal was to look at how the mechanics of this carotid artery changed as it filled with fluid. So if you filled the carotid artery with fluid, how did the structure of that wall change? If you add any kind of contrast agent into that specimen, it completely changes the mechanical properties of the very specimen that you're trying to measure. So that wasn't an option. So they had to use a different way of being able to capture that data. And they used propagation phase contrast to be able to generate the data sets. They could pick out the segment, they could segment out the layers. It was all done manually at this time to be able to look at the different layers of that muscle. And so using propagation phase contrast gave them the opportunity in the lab-based tool to be able to see something rather than not being able to do the experiment. So it's a very nice additional mode that you can run the system in. Thinking about biopsies, this is a skin sample here. Again, it's been prepared ultimately for volume EM in this case, and we were playing around with the different properties of the system using different objective lenses to really define exactly which combination of properties of the system would give you the best possible result. So on the left-hand side, this is a hair follicle here. This is the 4X objective lens. Large field of view, but you really don't get to see all of the details of the specimen, particularly because the region of interest was kind of down here. Being able to look at the morphology of the cell groups that you found here was what was most interesting to this researcher. And unless we pushed the system to the limits in terms of resolution and signal to noise, we just simply couldn't see it. So it wasn't until we reached the 40x objective lens and used something called deep recon, so using deep learning reconstruction rather than classical reconstruction, we were able to get a really good signal to noise ratio, and we were able to get to high resolution. And now you're able to start looking at the morphology and the shape changes that these cells would undergo in diseased tissue versus non-diseased tissue. So really at the limits of what you can achieve technology-wise. And similarly here, this time looking at a brain specimen. So this is mouse brain now. And we've used, again, the deep learning reconstruction tool here to be able to reconstruct the data. The cool thing about deep learning reconstruction is that it needs less data to input, and it can have noisier data to input. So you can capture your information more quickly, so your scan time reduces, and yet you still get an improvement in your signal to noise ratio. And this is just a two-dimensional slice through, obviously, the three-dimensional data set that we have of the whole slice. And we're able to pick out the processes of these neurons and start to look at how they are connected together. A slightly different scale now. This is a micro CT, so this is taken with the context micro CT system, looking at whole organisms, so the whole mouse embryo, whether you're interested in the toxicology of pharmaceutical agents, if you give them to the mothers, for example, or you're interested in the different genetic models of mice and how that changes their phenotype. All these kind of questions are accessible, and using X-ray technology, it's just a fantastic way of being able to see all the details of all the different organs inside that mouse embryo without physically having to section the system, having to section the specimen at all. The actual sample preparation process, just as an example, to create this kind of embryo phenotype library, you fix the embryo, you can then mount it in some hydrogel scaffolding that actually helps to prevent the sample from shrinking. The sample is then contrast enhanced using iodine, and then it's imaged using micro CT. And once you have that combination of images, the next challenge is what you do with that data. So on the left-hand side here, we have a two-dimensional section. So it's a three-dimensional reconstruction, and we've just sectioned it so that you can see the internal components in one part of it. We've then used deep learning reconstruction, segmentation, sorry, to be able to segment out different components. So the heart, the liver, the kidney, the neural structure, and also the eyes, in this case, of this mouth embryo. You can see those in the context of the rest of the mouse. So all the vessels associated with the mouse you can also see, and you can also pull out those different elements of that mouse. So you can start to compare things like liver size and volume, heart size and volume between different mouse models. And it's this combination of non-destructive imaging which gives you all this information, and actually what on earth you do with all that information. You can use deep learning tools now to be able to help to segment out and give you answers to those questions. In the world of vessels, this is a heart, a rat heart in this case, that's been imaged here. This researcher actually uses MRI technology a lot, does a lot of in vivo imaging with MRI, but just can't get to the high resolution of the vessels that she needs to be able to quantify the impact of some treatment or other. This is actually the rat heart that's suspended here in low melting point agarose. So it's in a falcon tube and imaged inside the versa. And we're able to look at those vessel structures in 3 dimensions. So the left-hand side is a 2-dimensional photo of the excised heart before it went into the system. And on the right, you've got a 3D reconstruction of that tomography data set, which is really amazing. And then in the world of diagnostics, so this is a really lovely preprint paper that came out last year, comparing how different patient situations can impact on the tissue. So this is a cancerous group and a non-cancerous group of human tissue biopsies that they've compared here. And the goal of being able to come up with predictive methods for diagnostics using AI. And the X-ray technology in this case was used. These are paraffin-embedded, unstained specimens, which have been imaged, around 80 of them in total, to be able to generate that deep learning matrix. Back to the world of mineralized tissue. So this is a whole fish in this case. As I said, bones are really difficult to image, can be very challenging to image in other ways. Using X-ray technology, you can reach high resolution insights in the specimen. The goal of this study was to look at how the bone changed as the fish were exposed to different swimming conditions. And they were particularly interested in looking at the osteocyte lacunae. But the really cool thing about the X-ray microscope, when you get to those higher resolutions, they didn't just look at distribution, you know, where they are, they could also look at orientation of those osteocyte lacunae. So the higher level information, high resolution information, gave them access to more data points and more comparative ways of being able to look at the fish relative to each other. Another really cool thing that you can do when you have that high resolution is to compare not only how a specimen looks, but also the mechanics of how it performs. And we have a lot of users using the X-ray microscope tools for exactly this. What you're seeing here on the left-hand side, you have a magnesium-based biomaterial, and on the right-hand side, you have trabecular bone. And the goal is to create a material that has similar mechanical properties to the bone. That's ultimately the goal. So what they've done is they've mounted their biomaterial and their trabecular bone inside an in situ stage, and they've then compressed, imaged the specimen to be able to generate a strain map of how the strain throws itself through each of the different specimens. So now you can bring together mechanical properties and morphology of what's happening within that specimen to give you even more information about your sample type. These are a couple of examples that I have for plant sites, which is just an amazing thing that you can do with the X-ray tools. Here we have a plant that's growing in the tube here, and we're looking in the first instance at the whole root and how that root grows through the soil. It will come in a minute, and you can see how the root grows, how big it is, how it branches, those kind of questions as it grows through the soil. What happens when you put different nutrients in the soil, for example. But using the X-ray microscope, you can then zoom in with higher resolution and start looking at how the microbes and the root are interacting together. So the goal here was to explore that symbiotic relationship, how the microbes are replicating and how they're interacting with the root. And using the 4X objective, we could pull out the different microbes and segment those out and look at that relationship. So again, it's the multi-scale element that was really interesting. But it's not just roots, it's also any part of the plant. For example, here we are looking at auxiliary buds here. And what the real benefit in plant science is the fact that you can get to these cellular resolutions, but within the context of that whole plant. You can see in 3D space where all these different elements are relative to each other. And that really is showing itself to be a useful additional imaging focus and imaging possibilities within the plant science world. So now on to the final section, which is talking about multimodal imaging. And Liz already did a great job of introducing this as a concept. So by the time you get to the synchrotron, or by the time you get to the electron microscope, you've invested an awful lot of time in your specimen, and you want to make sure that you can get the best possible results out. And many people are using X-ray technology as a screening tool to be able to assess sample quality before you get to a higher resolution technology like the electron microscope. And this paper is a great example of that. Yuxin imaged a lot of olfactory bulbs that she created ready for electron microscopy. And she found there are many different artifacts and weird things that can happen during the sample prep process. For example, you can get cracks in your specimen. You can have the staining that is only on one side for whatever reason. Sometimes the osmium never reaches the middle for whatever reason. And there were lots and lots of different variations in terms of sample quality that were not quite right, which would have compromised the electron microscope and the synchrotron results. So this paper was all about how you could use the X-ray, the lab-based X-ray tool to screen your sample for sample quality before you go onto the electron microscope. And it's kind of the standard practice for many labs now who are going to volume EM particularly to make sure you get the best possible specimen before you go. Another way of being able to use X-ray technology, this time actually live inside the system. So this is a 4mm cylinder of brain tissue that's been put inside the X-ray instrument inside osmium. So you have osmium going live into the specimen, and it was imaged in the end for a total of 56 hours. What you're seeing here are the two-dimensional projection images as that osmium kind of makes its way from the outside to the inside of the sample. And what this allowed them to do was to be able to benchmark exactly when the right moment was, when the osmium was absolutely at the center of that specimen. And it was, say, 56 hours. So again, another way of being able to use X-ray technology to optimize image quality. and of your final result with the EM. Optimization is one thing, but the other thing that X-ray technology is great for is identifying within a completely opaque block exactly which region you want to capture at higher resolution. So these are specimens that have been prepared for serial block face in this case. They've been imaged using the Versa, the X-ray microscope here, identified the region of interest, and based on the measurements from that specimen, you can then go on and use the ultramicrotome to section that specimen to exactly the right size before you go to the electron microscope. So checking for sample quality and then getting the sample to exactly the right dimensions so that you can get the best possible result from your electron microscope are both really, really important uses of X-ray tools. And in the zebrafish, another lovely example of this. So here we have the fish imaged in fluorescence here, but with the X-ray tool, you're able to identify exactly the region that you want to image here, the ear of the zebrafish. The same specimen is then imaged on the FibSem in this case. You can identify the region of interest, and actually using software, you can then point with the software to identify exactly which region you want to capture with the focused ion beam system. So it really takes a lot of the pain out of the workflow of getting from your bulk sample through to the higher resolution insights. So it's exactly the right place. And you then, with your fib, are able to capture all of the sections at super high resolution, and you have those sections within the context of the whole fish. And finally, again, looking at high resolution data, this is a nerve bundle here. It's a periphery nerve in this case. And their goal is to be able to look at the myelin across the whole length of this peripheral nerve. It's a very ambitious goal. These are really large specimens ultimately. But we're trying to use some of the newer tools from the reconstruction side to be able to assist with this. And this one is something we call Deep Scout, where you capture the whole field of view at a relatively low resolution, and you capture a smaller field of view at high resolution. And then the reconstruction parameters that you use for this region, you can apply across the whole larger field of view. So you can essentially pull higher resolution from the lower resolution data that you've captured. This enables you, so traditional reconstruction on the left-hand side, Deep Scout reconstruction on the right, you're able to look at things like the thickness of the myelin, how is that myelin affected across the whole length of that peripheral nerve? And it's these kind of jumps in the technology which are really assisting now with some of these super challenging projects and yeah, incredible science. So that brings me to the end. I hope I've at least given you an overview of some of the stuff that's going on with lab-based tools and how it complements the incredible stuff that's happening at Synchrotron. If anybody has any questions, I would be delighted to answer them, although I'm aware we have 1 minute before the end. So thanks very much.